Hello, this is Gender Wolf. In this video I'm going to be talking about the genetics of sickle cell disease. Now, in order to understand the genetics of sickle cell disease, we need to talk about how genes are passed on from mother to father. Now, sickle cell disease is an inherited genetic disorder that is a recessive trait. Okay, in order to understand how sickle cell disease is passed down, we need to talk about the process of how genetic material is created and passed on through the ova and sperm. Now, what I'm drawing here is the 11th chromosome. The 11th chromosome is important in sickle cell disease because it is where we find the mutation, the gene that is mutated, and it's the gene that forms the beta chain of hemoglobin. And this is the sort of point where we have a mutation that causes sickle cell disease. So we're going to talk about the 11th chromosome and how the 11th chromosome is passed on from mother and father to child. So here we have an 11th chromosome, and we'll say this is in the mother, and the mother has a gene that one half of the 11th chromosome was passed to her by her father, and the other half of it by her mother. Now, as this 11th chromosome is going to be passed on to her child through her ova, which is formed through the process of meiosis. And during the process of meiosis, all the chromosomes sort of get mixed up. So the 11th chromosome ends up being sort of chopped into bits and then reformulated, so it becomes looking like this. So part of this half of the 11th chromosome comes from the mother. So we're going to see how little bits kind of get mixed together here. So we have this is sort of chopped up into bits and then reformulated here. So we end up with the same 11th chromosome, but you can see it's been mixed and matched so that the father's side and the mother's side get remixed into another form. Now, this is going to get divided off. One half of this is going to get divided off to form what's called a haploid cell, which only has a half of each chromosome. So you can see that the 11th, this ovum now contains half of the 11th chromosome. Part of it comes from the father's side, and part of it comes from the mother's side. Okay, so this is one half of the 11th chromosome. Why is this important? Well, imagine that this mother, so this is the mother cell, and this mother, say she is a carrier for the sickle cell trait. That means that on the 11th chromosome she has a mutated gene, and we'll say that that mutated gene got passed along in this little piece of the DNA in the ovum. So an ovum that contains this piece of the DNA could be passed along to the child. So the ovum that contains this mutation will pass along the sickle cell trait. Okay? So this ovum that contains the piece of the 11th chromosome with the sickle cell trait on it will pass on the trait to the child. This one that doesn't will not. So a mother that has a one half of the chromosome that carries the sickle cell trait has a 50% chance of passing on that trait to the child. Now, this is a recessive disorder, and we're going to talk about what that means in a few minutes, but what that means is that only one copy of DNA with the sickle cell trait is not enough to cause the disease. It needs both copies. So we need to actually have the father present a sperm that also contains a copy So let's see. Let's change the colors here. So we've also got a piece of DNA, 1/2 of the 11th chromosome, and imagine that this aspect part of the chromosome contains the mutation for the sickle cell trait. Okay, so we've got the same sickle cell trait here that's being passed on by the father. So this person now has the same mutation on both sides of the 11th chromosome. Okay, and this having the sickle cell trait on only 1/2 of the chromosome will not cause disease, but having the mutation on both halves of the 11th chromosome will cause the disease. Why is that? Okay, in order to understand this, we need to think about what genes do, and they actually help. They are the protein. They are the blueprints for the creation of proteins in our body. So the 11th chromosome, one portion of the 11th chromosome helps create the beta chain of the hemoglobin protein. Okay, so what does this mean? It makes a polypeptide. So the gene for the beta chain of hemoglobin is made up of about 438 nucleotides that code for, I think, 136 amino acids that make up the protein. So there are sort of 3 letters, each 3 letters of nucleotides codes for a single amino acid. And for more information, actually Saul Kahn made a great video to understand this a little bit better. So they're sort of grouped up into 3. And along the 136 codons, or sets of 3 letters, there is one set of 3 letters that codes for, that is a GAG, and it codes for a glutamate amino acid to be added into this protein. And that is coded for by a string of 3 nucleotides that codes for glutamate. Now what happens with sickle cell disease is actually the adenine nucleotide is replaced by a thymine nucleotide. And what this does is the GTG changes the glutamate amino acid to a valine amino acid. And so this is a small but significant change in the makeup of the beta chain of hemoglobin. Okay, so a person that only has the trait, only has 1/2 of the 11th chromosome that has this mutation, will have half of the DNA functioning normally and producing the beta chain that has the correct glutamate in this position. So what's going to happen? Well, half of the hemoglobin that gets produced will be normal hemoglobin A because it has a normal beta chain. The other half will be producing hemoglobin S because it has an aberrant amino acid within the beta chain. So we know that patients with people with the sickle cell trait that don't have sickle cell disease actually are creating some hemoglobin S, but only about a little less than half, around 40 to 45% of their hemoglobin is hemoglobin S. And this is not enough to cause symptoms. We have to have most of the hemoglobin be abnormal for it to cause disease. So this is why it's a recessive trait. It's not that people with the trait don't have hemoglobin S being created. It's only that a portion of their hemoglobin that's created is hemoglobin S, and the rest of it is normal. So around 45% of it is hemoglobin A, and the difference is going to be other types of hemoglobin like hemoglobin F, which is fetal hemoglobin. So in order to have symptomatic disease, both sides of the 11th chromosome, so the 11th chromosome that's donated by both the mother and the father, are going to have this mutation that causes the glutamate to be replaced by a valine, so that all of the hemoglobin that is created becomes hemoglobin S, or the vast majority of it. So you end up with, 48% plus 45%, and you end up with the vast majority of the hemoglobin being produced. 93, greater than 93% is going to be hemoglobin S, and that will be sufficient to cause disease. To understand this a little bit more, watch my first video on the pathophysiology of hemoglobin, and you'll understand why you need a high concentration where the majority of hemoglobin in the erythrocyte needs to be hemoglobin S in order to cause disease. So I hope this was helpful to you, and I hope that you enjoy the rest of my videos on sickle cell disease, and I'll see you there.